I am pleased to introduce Penny Smorenburg. Penny is a support and applications manager at KAPA PROBE based in Cape Town, South Africa. She manages part of a global team focused on scientific support, customer collaborations, and product applications. Penny began her scientific career working as an intern at King Edward VIII Hospital in Durban, South Africa, where she was trained as a biomedical technologist specializing in virology. Before joining KAPA, she spent the majority of her career at Molecular Diagnostic Services in Durban, where she focused on developing human and veterinary molecular diagnostics. Her extensive experience spanning across pathogen detection, Sanger sequencing, and more has been an invaluable asset to KAPA, both during product development and to our customers when troubleshooting challenging problems. Please join me in welcoming Penny Smorenburg.
Thanks, Charles, for that introduction. Right, during this webinar, I would like to discuss three main topics. First of all, I will give a brief overview of Transgenics and its role in diagnostic applications. I will then move on to a discussion of various genotyping workflows in use today, and we'll then spend time talking about how KAPA Biosystems solutions for mouse genotyping and how we are constantly evolving our products to meet the demands in the marketplace. We will now start with an overview of the growth of transgenics in medical research. In 1980, Gordon and his team were some of the first to demonstrate that the technology for developing lines of transgenic mice carrying cloned genes was both tractable and reproducible. In 1984, it was reported that oncogene expression in normal cells within normal tissue could lead to tumor development in mammalian organisms. Others substantiated this hypothesis between 1985 and 1987. Later on, in 1992, gene knockout technology converged with tumor suppression methods. This allowed researchers to generate mice that would develop cancer because they lacked tumor suppressor gene function. All of these early technological breakthroughs led to the development of genetically engineered mice with heritability developed forms of disease. This then gave scientists the ability to gain a better understanding of a particular disease, both for the development of new medical therapies and to develop and to evaluate existing ones. As a quick refresher, the schematic shows two methods for developing transgene mouse lines. These are pronuclear injection, which is commonly used in expression, and transfection, which is most commonly used in knockout models. In expression workflows, transgenic mice are generated by pronuclear injection of foreign DNA into fertilized mouse oocytes. These oocytes are subsequently transferred into the oviduct of pseudopregnant foster mothers. Targeted mutagenesis is also known as gene knockout or knock-in. Here, embryonic stem cells are transfected with the targeted transgene. The transgene then undergoes homologous recombination with the wild-type gene. After selection, positive embryonic stem cells are introduced into blastocysts and implanted into foster mothers. Chimeric mice can be identified based on the mixed color of the offspring, and heterozygous mice are bred to obtain mice that are homozygous for the mutant allele. Now to move on to some workflows that are currently in use for mouse genotyping. As shown in the schematic, genotyping workflows all follow the basic route of sampling, lysis, cleanup, and analysis. Within this basic layout, there are a number of possibilities for each step. For sample collection, the nature and quantity of mouse tissue required for genotyping are based on the type of DNA test being performed. The massive amplification inherent in the polymerase chain reaction, or PCR, permits identification of the gene of interest using only a very small sample of DNA. Hair, saliva, tail cuttings, and ear punch biopsy snippets are generally sufficient. In addition, blood samples stored on FTA cards rather than in larger blood vials are often used. There are many options for lysis of tissues for analysis. Chemical lysis uses reagents such as alcohol, chloroform, sodium hydroxide, or EDTA, and is used to permeabilize the cell wall and membranes. On the other hand, mechanical disruption is also quite popular, although some methods are used more than others. The most common methods requiring instrumentation include sonication and homogenization. Manual methods such as the use of free thaw cycles or grinding with a mortar and pestle are also in use. All of these methods are simple and relatively low cost, barring the cost of equipment, of course, but samples will generally contain a high concentration of PCR inhibitors, necessitating some level of cleanup prior to PCR. In other words, lysis is only the first stage in the process. A DNA extraction of some sort is still required after lysis has been performed. Proteinase K is a broad-spectrum serine protease used in many DNA extraction protocols to digest contaminating proteins after either chemical or mechanical cell lysis. The main downside of this method, however, is the cost of the protease and the fact that there may still be a high level of carryover contamination. This is, however, dependent on the length of the digestion and which lysis method is used up front. Alkaline lysis, using SDS and or sodium hydroxide, is also a popular method, but must be followed by a neutralization step to make the material suitable for PCR. An advantage here is that it is quick, but the disadvantage is that the chemicals used for lysis are toxic, as well as being potent PCR inhibitors. Phenylchloroform extractions are used to provide high-quality DNA for molecular biology applications. Once DNA has been separated into the aqueous phase, it can be desalted and then concentrated using ethanol precipitation. This method is relatively inexpensive, but uses toxic chemicals and takes a long time to complete with many hands-on steps. There is also the possibility that phenol or ethanol may both carry through to PCR, so technique is paramount in this method. Solid phase cleanup is a silica or bead-based method whereby DNA molecules bind to a surface in the presence of certain salts and other certain pH conditions. Input material is lyed, washed, then eluted. Apart from the high cost of most solid phase protocols, there is also an inherent associated loss of DNA, all dependent on the method and the skill of the user. The method does, however, generate high-quality extractions, and the bead-based methods are automatable. In summary, although there are a number of methods for tissue cleanup, they all come with their own disadvantages and advantages. For this reason, it is important to consider your laboratory's capabilities when setting up a genotyping workflow. Many factors, such as expected number of samples, the type and number of assays to be offered, and the cost and time associated with these, must all be considered. In addition, further factors such as sensitivity of the assay or method to sample inhibition and DNA input variation should be considered. For example, fine-tuning a quantitation assay with variable inputs could be a challenge. The stability of the system should also be taken into account. What would you accept to be an acceptable number of failures, and how consistent are your run-to-run results? Do you have to set up on ice, or would you like to set up at room temperature? And last but not least, do you have access to both endpoint and real-time thermocyclers, or is this a determining factor with regard to workflow? We will now move on to the mouse genotyping options offered by KAPA Biosystems. But first of all, let's look at what sets KAPA's enzymes apart. Directed evolution is the method of protein engineering that simulates natural selection in the laboratory. The process starts with a gene coding for a wild type or unmodified enzyme of interest. Random variation is then introduced into the gene through a process of mutagenesis, creating a library of millions of genes, each coding for a unique enzyme variant. The functional selection pressure is then applied to the library, and only the genes that coded for the highest performing enzymes survive. The process of random mutation and selection is repeated until the desired enzyme function has been obtained. Since introduction of a random mutation into a gene is mostly harmful to the function of the encoded enzyme, unlocking the power of directed evolution requires the ability to screen very large numbers of enzyme variants. Our core technology combines a high-throughput emulsion format, which is made up of aqueous droplets suspended in oil that serve as microreactors, with high-throughput selection assays in order to enable the functional screening of hundreds of millions of enzyme variants in parallel. The majority of DNA and RNA modifying enzymes are wild type. This means that they are isolated from nature without modification, and were never intended as tools for molecular biology. Directed evolution enables us to engineer improvements to enzyme function, such as robustness, thermostability, selectivity, processivity, or resistance to inhibitors, all of which require global changes to protein structure. These functions are not amenable to rational design, which is a method of protein engineering that requires detailed information about enzyme structure and function. These evolved enzymes are thus different from other engineered enzymes because they're optimized for functions that cannot be addressed using rational design. The first products that I will discuss are the components of our KAPA mice genotyping kit, a crude extraction and endpoint PCR combination that allow for rapid throughput genotyping with very little hands-on time. The crude extraction method we offer is KAPA Express Extract. This is then paired with our KAPA2G fast genotyping mix, which is a second-generation enzyme mix specifically designed for crude PCR and which comes in both hot start and non-hot start formulations. As discussed previously, there are various methods of combining lysis and cleanup methods to generate PCR-ready extracts for genotyping. When comparing the various methods, however, it is obvious that the methods that provide the highest purity DNA also have the highest time and cost associated with them. Lower cost methods may be quick, but will leave a higher concentration of inhibitors in the samples. KAPA Express Extract strikes a balance between cost and time, and will be discussed in more detail during this presentation. It should, however, also be noted that direct PCR is on the schematic. This has both the lowest cost and time implications. Direct PCR is clearly not an extraction method. It is, however, a goal for fast throughput workflows, but it's seldom possible because of the tissue or blood inhibitors taken into PCR. Direct PCR will also be touched on later in this presentation. KAPA Express Extract is a novel thermostable protease and buffer system that allows for the extraction of PCR-ready DNA in a thermocycler or on a heating block in 15 to 20 minutes. A small amount of material is added directly to the lysis buffer, and the tube is then incubated at 75 degrees Celsius for 10 minutes, then inactivated at 95 degrees Celsius for 5 minutes. Thereafter, the tubes are centrifuged to pellet the debris, and 1 microliter of neat or diluted material can be added to the PCR without further cleanup. For mouse genotyping endpoint workflows, we recommend the use of KAPA2G fast genotyping mixed with dye, which I will cover in a few slides' time. In addition to its ability to rapidly extract DNA, KAPA Express Extract is extremely versatile across sample types and produces high-quality DNA. Mouse genotyping workflows commonly begin with tail clips, ear punches, blood, or hair, and all can be extracted efficiently with our kit. These samples can also be taken to standard Sanger sequencing, as in the example shown here for species identification. Express Extract can also be used to prepare PCR-ready DNA extracts from other sample types, such as FFPE blocks, although these are not commonly used for mouse genotyping. KAPA Express Extract is not the only part of the KAPA workflow solution that helps streamline protocols. 2G fast DNA polymerase, part of the mouse genotyping kit, was engineered through directed evolution for extreme speed. Compared to wild-type enzymes, as shown here, KAPA2G can reduce cycling times by up to 75%, even for long targets. At times, you may have limited starting material, either because samples must be used for multiple experiments or targets, or because of variation in sample collection or extraction yields. The typical approach here would be to increase cycling extension time or to add additional cycles to ensure sufficient amplification. With KAPA-2G fast, speed does not, however, come at the expense of sensitivity. Even at 1 kb per second, the 626 base pair target can be detected down to 10 picograms in only 35 cycles. When used independently, both Kappa Express Extract and the KAPA-2G fast kit are capable of accelerating either extraction or PCR workflows. When combined, they offer an elegant, streamlined solution that enables PCR in as little as 1 hour for mouse genotyping. Other commonly used workflows for mouse genotyping can take more than 4 times that, showing that the Kappa mouse genotyping kit allows for dramatically increased sample throughput. In addition, and as discussed before, the Kappa mouse genotyping kit is highly optimized and maintains the versatility of its individual components. In this example, amplicons ranging from 312 to 915 base pairs with a GC range of 49 to 65% are all efficiently amplified, not only from genomic DNA, but also from tail, ear, and toe samples. Because crude extracts are not generally known for their ability to amplify longer targets, we tested a tail extracted with Kappa Express Extract against purified mouse genomic DNA. Although purified material performs far better, up here at the top, and can amplify up to a 5 kb fragment using the 2G fast hot start genotyping mix, a 1 in 10 dilution of our crude extract was able to efficiently amplify a 2 kb target. When working with crude extracts, there is often competition between the inhibitors and the PCR product, especially when working with more challenging targets. As can be seen here, a 1 in 10 dilution of the Express Extract product provides a far cleaner result because removal of the inhibition allows for more efficient PCR. We recommend this dilution in our technical documentation as best practice. In summary, Kappa offers an endpoint genotyping solution consisting of our Kappa Express Extract and our second-generation genotyping mix. Processing from extraction to gel image can be done in approximately an hour and a half, depending on the size of the product. It's important to bear in mind that about a third of that time is the time taken to run the gel. So far, we have used our kit with tails, ears, toes, and blood, all with excellent results. In addition, we've found our mix to be robust over a wide range of products, both with respect to size and GC content. I will now move on to our exciting new qPCR offering for mouse genotyping, namely KAPA PROBE FORCE. Probe FORCE contains our third-generation enzyme, Kappa-3G hot start DNA polymerase, which is specifically designed for robust qPCR. With this kit, we are able to offer an even more streamlined sample-to-result workflow. The reaction mix allows for both crude and direct qPCR, while offering superior inhibitor tolerance and accurate, reproducible, and sensitive results, either in singleplex or multiplex formats. When developing this reaction mix, a dilution series of purified mouse genomic DNA from 100 nanograms to 100 picograms was run against inhibitor-spiked mouse genomic DNA. Using the purified 100 picogram purified sample as a baseline, we took a look at how significantly CQ values shifted with 5 tissue inhibitors common to mouse genotyping workflows. KAPA PROBE FORCE showed the smallest delta CQ values when compared against 5 competing master mixes, all of which have been advertised as being robust mixes for qPCR. In some cases, over here with the pink NA, no amplification was seen, not only at 100 picograms, but across the full dilution series. In others, the pink 100 nanograms, a much higher DNA concentration was required for amplification to take place. Highly processive enzymes are required for efficient multiplex assay performance, and finding optimal reaction conditions suitable for all targets can present a challenge. In this example, KAPA PROBE FORCE with minimal assay optimization was able to efficiently amplify all 4 targets in a multiplexed reaction. In addition, it resulted in early PQ values and a higher final fluorescence than competing master mixes. This is when used with the recommended fast cycling conditions, which were similar for all mixes tested. We then compared our kit to DNA extracted by 3 additional methods: 2 semi-crude methods, namely sodium hydroxide and proteinase K, and 1 column-based extraction method, the QIAGEN QIAM's DNA mini kit. The combination of express extract and probe force offers significant time savings and a simplified workflow, just like our mass genotyping kit. However, we wanted to confirm that this did not come at the cost of performance. Using tail, ear, toe, and hair samples of both neonate and hopper mice, as shown in the table, we measured expression levels of beta-2 microglobulin, which is a single copy region of the genome, with the KAPA PROBE FORCE qPCR kit. Neonates and hoppers were both shown to be suitable templates, with the neonates performing marginally better overall. For tail, ear, and toe templates, all extraction types produced yields of greater than 10 nanograms per microliter, which is the dark blue line over here. Hair samples yielded concentrations of around about 1 nanogram per microliter and slightly lower, which is measured against the pale blue line. Furthermore, all reactions had high reaction efficiencies and low replicate variability, regardless of sample type or extraction method. When comparing the extraction methods, KAPA Express Extract resulted in higher yields with less input materials and column extractions, was equivalent to sodium hydroxide preparations, and was slightly less sensitive than proteinase K. None of the reactions showed any signs of inhibition, and all reached the same final fluorescence as the purified DNA controls. In addition, probe force is capable of direct amplification from tissue and blood spots. For this experiment, we used 6-year-old FTA blood spots and ear punches from neonate and hopper mice. To demonstrate the effective sample size when using a 0.5 ml punch, 1 or 2 ear punches and 1, 2, or 3 blood spots per reaction were compared. Regardless of number, all of the blood spots produced consistent results, yielding slightly less than 1 nanogram of DNA. This highlights the fact that increasing the amount of blood inhibitors in the reaction mix does not adversely affect the quality of the reaction. In addition, sufficient amplification can be achieved from very low inputs. All ear punches produced yields of around about 10 nanograms per microliter, again with the neonates performing marginally better overall. The important thing to note here is that direct PCR may not necessarily be possible or even the optimal testing method for certain assays or determinations, especially those such as quantitation. However, it is definitely possible and would be best suited to yes, no answers in the laboratory. To represent the extremes of what might be received, tails were cut to sizes of approximately 3, 5, and 10 millimeters, and the extract was then run neat on a 4-plex assay. The 5 and 10 millimeter extract showed no signs of inhibition, instead performing slightly better than 3 ml extract. This slide serves to show the sensitivity of the probe force qPCR master mix in a multiplex setting, as well as the robustness of the mix with regard to input. To demonstrate the stability of the reaction mix for high throughput scenarios, the reaction mix, excluding DNA, was incubated at room temperature for up to 3 days. The tubes were kept protected from light during the incubation, after which 10 nanograms of mouse gDNA was then added to each tube prior to qPCR. As can be seen, the reaction mix shows itself to be highly stable in the absence of template. We then did the same with our multiplex assay, which showed great stability in the absence of template. To take this a step further, the full reaction mix, including either mouse gDNA or a neat or diluted express extract template, was incubated at room temperature, protected from light, for up to 4 days. A T0 sample was used as a control, and the 4-day aliquot was also kept at 4 degrees Celsius. The reaction mix shows itself to be highly stable in the presence of both genomic and diluted express extract templates, but the neat crude extract shows signs of drop-off within a day. However, 4 days at 4 degrees showed no drop-off in any of the templates, indicating that this would definitely be a viable option for high-throughput laboratories. We did the same again for our multiplex assay, and again, the genomic and diluted material proved to be very stable in the reaction mixes over a 4-day period. From this presentation, it is clear that the combination of KAPA Express Extract and KAPA Probe Force provides a fast genotyping workflow while using crude extracts. Probe Force is also compatible with other extraction methods, providing high levels of inhibitor resistance for those methods often associated with carryover PCR inhibition. Not only does KAPA Probe Force show consistent performance, regardless of tail length variation, but it is also capable of direct amplification with no loss of efficiency. Finally, the fact that the reaction is stable both with and without templates for a 3 to 4 day period means that this kit is well suited to both low and high throughput laboratories. So in summary, transgenic mice are essential tools for research into human disease. Screening of mouse populations can be time consuming, and genotyping workflows should always be able to balance time and assay performance. With KAPA's endpoint and qPCR mouse genotyping offerings, we're able to provide high-performance solutions that are both time and cost-effective without sacrificing quality. Thank you very much for listening to me. Do you have any questions?
So one question is, what are the most important parameters that must be considered when attempting to convert an assay from purified to non-purified samples?
Depending on whether you're working with endpoint or qPCR, this can have two different answers, so I'll go through both. With endpoint parameters, with endpoint PCR, the parameters that would most be needed would be to look at increasing the cycling time, very possibly, or the number of cycles, should I say, and also, once moving to crude, working with a 1 in 10 dilution. We have found that certain assays with a neat do not work as well, and it definitely seems to be related to certain primers, certain targets, but a 1 in 10 gives a very good, consistent result throughout. When working with purified, we would recommend about 35 cycles, and working with crude, we would recommend going up to 40 cycles for PCR as a first step approach. For qPCR, especially using Probe Force, you would have to do almost nothing different. 1 in 10 diluted and neat both work almost the same as purified DNA. So as long as you're working against a standard curve and you test it a few times, you'll probably find that you won't have to change any parameters going forward.
Another question is, have you tested the compatibility of Express Extract with the next-gen sequencing?
Good question. At the moment, I would say that we would not recommend it going forward to next-gen. We definitely know that it is good for Sanger, but taking it to next-gen at this time would be something that we would say, if you want to try it, you're welcome to, but we cannot guarantee the quality of the DNA. So because of the fact that it's a crude extraction, even a diluted template going through might not be pure enough to allow for next-gen sequencing.
Another question is the contents of the extraction buffer proprietary? Yes, they are, and as is our other evolved enzymes for each of our products. Those are all proprietary content. All right, well, if there aren't any other questions, thank you all again for attending. We appreciate your time and hope you've learned some new methods and techniques on how to optimize your mouse genotyping workflow and overcome PCR inhibition. If you have any questions that come up, please email our support team or our sales team if you're interested in testing any of the products that we've presented today. Thank you and have a good evening.